No? It's there. Իստումա մենք առանց չէ, լավ իրաց լսում ենք։ Հիմա առաջի հերթին բարև ձեզ, շնորհակալություն, որ դուք գտել եք ժամանակ եւ չի լսվում։
Չէ, որ արձակությունը հեշտ լինի։
Հա, ձայնագրություն համար պետք է։ Լավ, օքեյ։
Hold it up.
Right, somewhere there.
Oh, hold it.
Can you hear me? No?
Չէ,
And now? And now? No? One more? Oh, yeah, it's there. Շնորհակալություն, որ դուք ժամանակ գտաք եւ կարողացաք գալ այսօր։ Հիմա առաջի հարցը, որը ես կուզենայի ձեզ տալ, դուք ինչ լեզվով եք նախընտրում ես խոսեմ։
Անգլերեն։
Անգլերեն։ Վստահ եք։ Լավ։
Ինչ լեզվով
Չէ, ինձ
Technical
միանգամայն մեկ է, ես կարող եմ խոսել հայերեն technical terms անգլերեն, ես կարող եմ խոսեմ անգլերեն technical terms անգլերեն, ես կարող եմ խոսեմ հայերեն, բայց technical terms-ը ամդեքում անգլերեն ալին է, ուտես յանց հայերեն տարբերակները չգիտեն բազապես։ Okay, so what I want to present today is not based on my sole research, but more on it's an introduction to what we do in Armenia, what kind of research is done in our laboratories, why we do that, and what we get as a result of our research. So, but the first few words about the institute. So, institute is relatively young. It has been established in 1966, and it was called Institute of Experimental Biology. It has been established from several groups that had mostly biochemistry and physiology background, because they came from Institute of Biochemistry and Institute of Physiology. So, though it was called experimental biology, what they did, they mainly work in biochemistry and physiology. The first director of the institute was Dr. Stepan Chishmaityan, and as far as I remember, he was from US. Institute has changed about 6 directors, but the most influential person in this position was Professor Anna Boyajan. She has almost all her scientific career was connected to the institute. Finally, in 2006, she became the director of our research center. And we consider this 2006 as a point from which life has research in, it's much better. It's too loud. Okay, let me continue in this way. So from 2006, new age of our, let's say, development began. What she did, she was a woman of science. She was distinguished researcher, and she was a very good administrator. She was the one who realized that it's time to switch from the old, outdated methods to new, modern research approaches and to new, modern, contemporary, and well-recognized research areas. So here I just pointed some of the most influential actions that she made for our institute. This was switching from pure biochemical research to molecular biology research and genetics. She established and helped a lot of labs to find out the way and to discover new research areas that were interesting and also fit to our capabilities. Because you know that after 19th of 1990, the war, the independence, it was a very hard period in Armenia. A lot of scientists left, financing was very low, and the most enthusiastic people stayed there. But they were needed some guidance, and she provided this guidance to that people, so they again restarted their scientific career after long gap or delay. She was very keen to attract young scientists to the institute. This was one of her main administrative, let's say, duties or policies. She very well knew that young people at the research center is an engine, and they should be there. She also was very attentive to career opportunities. So she tries to set up new research units to provide opportunities for young scientists to become group leaders, become senior researchers. And also she helped a lot in forcing people to publish in international journals, to somehow overcome this feeling that the research they are doing couldn't be published in normal papers. So she really helped a lot of people to understand that what they do is really a good science and can be published in very good papers. And also she built a huge collaborative network. And we now use these results to keep the level on which we are now and try to extend it to more beyond that limits. So, what we do now? What is the research focus of our institute? We mainly are interested in understanding the mechanisms of cell activity in health, and of course how they change in different diseases, in humans as well as in animals. Traditionally it has taken direction to immunology, and we are still very focused on immune system as well as signal transduction systems that guide immune responses. Very new area or direction we started is characterization of Armenian genome, based on pathological, pathogenetics, echogenetics, immunogenetics, and also population genomics, to understand from where we had originated, how we evolved or developed, and if we really are predecessors of the people who lived here for a long period ago. Another important thing is educational activities. We really realize that without new generation which will work in our institute, we can't do much. And we try to provide young scientists at our institute with research-oriented degree programs that will make them good specialists and also help them to build their career further on. Now a few words about structure. The institute has very flat structure. We have simply administration and me as the director of this institute, together with scientific council and young scientist council. Administration decides prospective research areas. It defines more or less direction of research, and it then gets reporting from the head of labs, and so it coordinates the research activity of the institute. We have about 9 labs, 9 laboratories, and they work in very different areas. So you can see here genomics, cell biology and virology. Actually you can't see, but I will tell. Computational modeling of macromolecules, ethnogenomics, so everything which is interesting and which is possible to do at the institute. We have 6 groups, and all of them has leaders which are below 40 years. So they are very young, and this is specific policy at the institute. Once per 2 years we announce competition for young scientist research groups. So we get proposals, and then we select the best proposal, and this young scientist gets his own group with some staff, few people, and some resources. And we also have 3 educational departments which are established jointly with universities. So Department of Bioinformatics has been established jointly with Russian Armenian Slavonic University. Department of Molecular and Cellular Biology has been established with International Science and Education Center of Academy of Science. And PhD degree programs, we constantly have them because it was inherited from former Soviet Union educational system where research institutes should have had PhD positions. So this is our regular degree program, and we offer programs in molecular and cellular biology, genetics, and biochemistry. We also have several research units which are supporting units which are shared by all laboratories at the institute. First one, and the most important for us at the moment, is Genomics Research Sequencing Center. We have, by generous gift from ARPA sequencing machine, and we use it for in various applications, starting from simply analysis of different nucleotide sequences, finishing with validation of site-specific mutations. We have DNA sample bank. We constantly collect new clinical samples from healthy people and diseased subjects. At the moment, we have a bank for patients with ischemic strokes, with hemorrhagic strokes, diabetes, schizophrenia, post-traumatic stress disorder, and so on. We also do have 19 animal and human cell lines and small animal facility. And everything is used by all our researchers when it needed. Our staff is very young. You can see here that the most, if we take age categories, the most of our staff is below 35. These are newcomers, students, freshmen, second, third year students who are really interested in science, and we never say them no. So we always welcome everybody who wants to do science.
So are they volunteering?
They start with volunteering for 2 or 3 months, and then if they deserve it, we just give them position. This is the only slide.
How many PhDs you have? How many PhDs you have?
In our institute, we do have 24 PhDs and 13 Doctor of Science.
Candidates.
PhDs are candidate of science and 14 doctors.
Out of how many?
100.
Of 80, I'm sorry, I said 23? No, sorry, 43. Out of 80.
You say 130?
100 scientific and 30 technical staff.
Oh.
Of course, from this 100, some of them are part-time, so they work also in university or somewhere else. This slide is the only one I would a bit complain because it's about our funding. So in Armenia, there is one main instrument for funding the research institute. It's called basic financing for state institutions to preserve the infrastructure. Under infrastructure, staff is also considered. So mainly, what you can see here on chart, you see this big blue part of the pie. This is about 74–75% of this funding goes to salaries. The rest we need to somehow divide between paying electricity bills, heating, reagents. So basically there are almost no money for research, and that's why we are very active in obtaining other grants from everywhere, from all possible donors. So in Armenia there are also some competitive grant projects, and we are very successful of getting them. They are not very big, about $30,000 for 2 years, but we have almost 15 of them for 2015 and 16. Our young scientists also Usually they get a lot of awards during their career. So in 2000, this period of these 2 years, they got about 15 different awards for publications in impact factor journals, for conference travel, for organization of scientific events. And yeah, and if you have questions, do not hesitate to interrupt me and ask, okay? We have 4 big projects. Mainly they are funded through International Science and Technology Center, 2 of them. These are about $500,000 projects for 3 years. And we have 1 project funded by European 7th Framework Program, and 1 big project funded by Volkswagen Foundation of Germany. And I'm sure you know about ANSEF Foundation, Armenian Science and Education Fund. Yes. This year we got 5 grants from them. These are small ones. Usually our young scientists start their training of proposal writing with application to ANSEF. And they are very glad when they get this. Actually, budget is very small, $5,000, but they're really happy to get this first, their own grant money. We have enough equipment at the moment to do a good science and publish in relatively respected journals. I put it in order of maybe importance. So first one is the sequencer, which we use now actively, which was donation from ARPA Institute. Then we have quantitative PCR machines, freezers, gel documentation systems, different type of spectrophotometers, high-performance liquid chromatography device, and these all are provided either through gifts from different organizations or researchers, or through were purchased in framework of grant project. We do have quite a good collaborative network. They mostly located in Europe because simply it's closer to travel. But here we collaborate with University of San Francisco. Recently we started our joint project, writing joint proposal with University of UCLA. And also I had several meetings here with possible further development in University of Southern California. So I really try to more integrate our institute into American research area and had more close ties also with researchers from US. These are our publication statistics for 2015. We published 56 papers, and from them 43 were in international journals. And we had a lot of we traveled a lot and participated in a lot of conferences. We also had 2 book chapters published in international, let's call books. So we really publish a lot. These are our books. You can see them in the, is it right? Right? Yeah, these are 2 books related to anxiety disorders. This is post-traumatic stress disorders, genetics of post-traumatic stress disorders. A second book was dedicated to genetic predisposition to schizophrenia in general, and we published a chapter about this in Armenian population. These are journals in which we had published papers in 2015. You can see there also Nature, PLOS, Frontiers. So unfortunately not all our papers are there, but significant part we published in very well respected journals.
Do these books generate income?
No. These books do not generate
You
income.
give it out free?
Yeah.
They're like proceedings.
No, they are books on dedicated topic. It's not proceedings.
Why are they given away for free?
Because this is publisher policy.
When you have just a few chapters when you contribute only a few chapters?
In some cases, we even pay for that. It's
Yeah.
called open access. So publisher has policy, so to make it more accessible to more wider audience, it asks author to pay publication fee, and then he distributes these books for free. So it's more science, less business maybe. So this part, I'm really sorry, I would need to go in some details, because this would be small descriptions of projects implemented in different labs. And you can see we do a lot of different stuff, sometimes completely unrelated, but this also from another side, it's widen our opportunity to find good links, good collaborations, to make interesting research.
What is the extent of collaboration with the other institutions that you were mentioning, like UCLA, US?
Okay, so at the moment, we with UCLA and with UCSF in parallel write two proposals for it's so-called NIH
National
program.
Institute.
Yeah, National for
Institution of Health.
Yeah. There are specific calls. The mechanism is gone through Fogarty International Center, and it's the aim to develop the collaboration between American scientists and scientists from low and middle income countries. So we write two proposals there. One is related to schizophrenia treatment efficiency, and the second is devoted to testing of new chemical compounds we synthesize as neuroprotectors in ischemic strokes. But let me start with this. We have 7-floor buildings. Labs are located from 7th to 3rd floor. So I ordered descriptions starting from the 7th floor, and we will just go downstairs. So the first big laboratory in the institute is called Laboratory of Cell Biology and Virology. I would actually change the places because it's more virology than cell biology. They are very focused on study how of the pathogenesis or mechanism of viral infections, how they affect the cell, how they penetrate into a cell, how they replicate, and all the stuff which accompanies viral infections. They have chosen African swine fever virus. It's called African, but in 2007 we had a huge outbreak in Armenia, and virus penetrated from Georgia. And in Tavush region, almost all animals died, and the ones that even were not infected, they were slaughtered because there are no any treatment for this virus at the moment. So they try to understand the mechanisms of viral infection for this ISFV. They also are interested to investigate possible reservoirs, because virus is circulating in Armenia, and we need to know what are the hosts or how they are transmitted. And also they are very interested in study of genomics, genetic of that virus. Why it is interesting? Because in Africa, this virus circulates from domestic pigs through ticks to wild boars and then back. Wild boars are resistant to this virus in Africa. In Armenia, wild boars died with the same rate as domestic pigs. And moreover, in Armenia, there were no ticks. So these ticks were not identified. So there are no cycle really in Armenia. But still, virus is there. And they propose a hypothesis that virus may evolve somehow or change somehow to adapt to this new microenvironment. And they performed sequencing of viral genes. It's just a part of the data they collected. And they were able to show that in outbreak of 2012, the genome or genes in this virus has been changed a bit. they discovered that there are new intergenic region which was absent in virus that was isolated in 2007. Yeah.
Can you describe this chart? What is the
So
x-axis?
the chart above, these two charts are results of sequencing. So what is DNA?
What's
DNA,
the x-axis?
huh?
What's the x-axis?
X-axis is position of nucleotide in gene, in the sequenced part, and Z-axis is intensity of signal. So using these charts, we can reconstruct the sequence of DNA. And after we reconstruct it, we simply align them and can find parts which are similar and parts which are different. So on the lower picture, you see green parts and red parts which like sit one above the other. And there is a gap in 3 rows, but there are still some sequence. Maybe I can use my mouse. So you see here is a DNA sequence which presents in one virus from 2012, but it is absent from other viruses which were isolated. This means that this virus may change somehow. Why it changed and what it does, we don't know at the moment, but still it's just preliminary steps. Now we write another grant proposal with colleagues from University of Connecticut and Pennsylvania, which is dedicated to more detailed investigation of these viral genomes.
Have they tried to match that insert with any other organism, like any other virus?
Yeah, they have very interesting theory about horizontal transfer of DNA across species, and they try to they now really play a lot with these sequences to find out from where this intergenic part has came. Okay, this group is formed in 2016 as a part of our Young Scientist Group project. The head is Hovhakim Zakharyan, and because he was a postdoc in laboratory of Zaven Karalan, he also decided to continue his research in field of viral defense, and he called his group Antiviral Defense Mechanism, a group of antiviral defense mechanism response research. So what they do, their main research focus is to discover novel molecules or chemical compounds which will possess strong antiviral effects, specifically against African swine fever virus, because as I told you, there are no effective vaccine mechanisms for that. So they do a lot of research in this area using contemporary methods and approaches, and they have found that there is a plant-derived compound called apigenin, and it possesses significant antiviral activity. So it can lower the viral yield for 1,000-fold. So it's huge. And if it's applied on the very early steps of viral infection. And they now continue in this direction to understand whether this compound can be used as a part of therapy for animals during the infection stage. A laboratory of molecular genetics is led by Jean Nakatsoyan. They are interested, they are mainly do microbiology, and they are very interested in disease mechanism of Salmonella infection. Salmonellosis is actually a real problem in Armenia, and what is most problematic that a lot of strains has developed multi-drug resistance, so it's very hard to cure them. They made a research, they screened a lot of different strains for Salmonella, and what they find out that depending on the serotype of the bacteria, let's say, one type possesses very strong multi-drug resistance, another is much milder. So another can be cured using different combinations of antibiotics. And they were interested whether this is somehow related to genetic transfer of antibiotic resistant genes. and they discovered that this is, in fact, it has connection. They showed that in this is specific genetic structure called class 1 integron, which usually contains cassettes of genes which somehow which related to antibiotic resistance. And they demonstrate that in one serotype of bacteria, there are a lot of, the percentage of having bacteria having this cassette is very high, while in other serotype, other type of bacteria, there are almost all of them do not have this genetic structure, so they are prone to antibiotic, or they can benefit from antibiotic treatment. And they also conclude that prevalence of this genetic structure among bacteria may also somehow predict possible multi-drug resistance development. A group of plant genetics and immunology works with plants. They are interested, actually their main goal is to preserve endemic plant varieties, especially grape, in Armenia. You know that before the collapse of Soviet Union, there were about 840 different varieties of grape, and now in the deposits only 44 remain. There were huge loss of these varieties, and they now try to somehow genetically characterize these varieties and maybe develop mechanisms for preservation of these endemic grape types. So they try to characterize grape pigmentation, grape color on genetic level. There is several genes which are orchestrated by another gene, which is called MYB, and depending on which genes you will have in your, which mutations in these genes you would have, you will get different colors of grape. So they using different molecular genetic techniques, they have found different combinations of these mutations and derived so-called haplotype. Haplotype is combination of different mutations which often meets, gathers together. So, and this is called haplotype. So they derived several haplotypes and characterized these haplotypes on functional levels. So how they are related to colors of the berry in the grape. This laboratory is temporarily headed by me. This laboratory is the Laboratory of Human Genomics and Immunomics. Actually, I worked there for almost 10 years, but then I started my own group. But because Professor Bajan tragically and unexpectedly passed away, so now I'm taking care of her laboratory until someone will be able to take her place. So what we did, what we do here, we are interested in study how genetic background and differences in our genome associate with disease development and progression. We study genes of immune system, genes of neuronal development, genes of endocrine system to find which genes and which mutations in that genes can be biomarker for predicting of risk of disease development. And also we study how different compounds, natural or synthesized, may be used for as a protective therapy against diseases. Again, because we are here and I would like to present the result that we obtained using sequencer, I'm just simply a few images about research on neurotrophin receptor gene, genetic variability in schizophrenia patients. So we have chosen part of gene on exon 15, which is very variable, and we sequence that part, and we we were able to detect common mutations which are characterized already, but we also found some new mutations which were not reported before us. Now we need to replicate this study again. There are just examples for 5 samples. We need to replicate this again, and if we would complete it successfully, we will submit this new information into corresponding gene banks. Group of cell culture technologists headed by Nelly Babayan are interested in development of test systems to study how neurotoxicity affect regulation of gene expression. And also they study different antioxidant, anti-cancer, and immune system modulating compounds extracted from plants. What they have found that mycotoxins, which are very commonly can be found in corn, these are specific fungi, and it synthesized very toxic compounds. One of them is ochratoxin A. This ochratoxin A may affect neurons, and this affection caused by demethylating DNA. So DNA has two, like it can be in 2 states, methylated and demethylated, and depending on state, DNA can be active or silent. So ochratoxin clears these silencing marks and makes a lot of genes active, and these genes may also harm the neuron. So this is preliminary data, and they continue to study which genes are affected by this demethylation process. They also isolated about 26 different plant extracts and checked whether they are toxic for organism or not. From these 26 different plant extracts, they have chosen 4 which were non-toxic, and then they studied their antioxidant activity. They found out that several extracts from pepper shows the best antioxidant activity, and because it contains a lot of
what kind of pepper?
Huh?
What kind of pepper?
Spicy.
Spicy pepper, yeah. And there are a lot of different plant-derived chemical compounds like terpenoids, phenolics, and others, which really, and their concentration, with the increase of their concentration, they observed increase of cell protection against radical action.
So what's their model? They're using tissue culture?
They used cell culture. Actually, they used HEP. Yeah, HEP cell culture. Laboratory of Regulation of Cellular Activity focuses on investigation or study how lipids are modified and how lipid-connected signaling is modified in human peripheral blood cells during cancers. And also they try to study whether genetic mutations in enzymes, in proteins of this signaling cascade can somehow be related to breast cancer. And their research is very interesting. They are still on their way, they're collecting more samples, but they were able to show that on the very early stages of cancer, they can see very similar patterns of these changes in lipids. Lipids are fats that cover cell from the outer side. It's called plasma membrane. And modifications in this plasma membrane have very similar patterns almost in all cancers. Now they're collecting more samples to show that they are really different from the healthy cells, and maybe this can be at some point used as a biomarker for early detection of cancer.
Excuse me.
Yeah.
Who are they?
They are laboratory of regulation of cellular activity in Armenia, in my institute. Next is a group of immune response regulations. David Povozyan and his colleagues study mechanism of regulation of innate immune cells during autoimmune disorders and rheumatoid arthritis. What they do now, they want to understand whether serum of rheumatoid arthritis patients contain factors that may drive apoptosis of immune cells. And they have found that the serum really contains some pro-apoptotic compounds that drive these immune cells to their death. It's just very beginning of their study. This was the information he provided me. Another group of immunology, we have 4 of them. It's called molecular and cellular immunology, and they are specifically focused on familiar Mediterranean fever. You know this disease. It's also called periodic disease. It's also called Yerevanian disease. It affects 2% of Armenians in Armenia, and also it has estimated that each the carriage rate, not the disease rate, but the carriage of these mutations is about 1 per 20 persons. So 1 of 20 Armenians has mutations that may then lead to development of this disease. So it's very urgent issue for Armenians, and a lot of effort is spent to study this disease. Genetics is more or less, we know a lot of things about genetics of this disorder. We know gene, we know mutations that cause disease, but we really don't know how it happened. We don't know mechanisms. And this group specifically studied the mechanism of the disease, how disease is developing, why attacks or these inflammatory attacks happen in recursive ways. So you have attack and then it relapses and then again and again. So it's why it's called periodic.
Do you have a bank of the people who have this disease or something?
Yeah, we have not very big sample, blood serum collection from these patients. And also they are interested in autoimmune diseases like antiphospholipid syndrome, because in some senses they resemble each other. They have, not to say very similar, but similar mechanisms, but still they are also very different. I will directly proceed to familiar Mediterranean fever. So what they discovered that neutrophils in this disease, these white immune cells, which are non-specific to nothing. So it ate everything. It's the first line of the defense from the immune system. They, during this disease, they undergo spontaneous death. And we consider that this is a mechanism which allows these periodic attacks to relapse. But most importantly, she also demonstrated that the colchicine, the only drug that can be used during this disease, in very high doses may provoke attacks. Because when you supply high doses of colchicine to the patients, it activates inflammatory reactions of the cells. So it may provoke or increase attack frequencies. And this was first time shown by this group.
But originally that same drug is used as
this drug is
curative.
used, now this drug is used to prevent attacks because it destroys these neutrophils.
But originally when you say in overdose, what was it used for?
Sorry?
When you said if it is used in high doses.
Again, in FMF, there are a lot of patients that tolerate this drug. So drug is not efficient. And one way to overcome this tolerance is to increase dosage. And when you go to up some level, this drug can itself provoke attack.
Colchicine is used for gout, right?
Yes, also for gut. It's actually, it's not a treatment for familiar Mediterranean fever. It's universal way to kill a lot of immune cells, does prevent attack. It simply treats symptoms. It doesn't cure disease. That's why patients should take colchicine throughout their life.
It should or should not?
Should. Otherwise, they would again start to
Recur.
experience attacks. So, Laboratory of Ethnogenomics, headed by Professor Levon Yepiskoposyan, is interested in genetic history of Armenian population. He tries, based on different markers on Y chromosome, on the mitochondrial DNA, or on the whole genome level, to show the continuity of Armenians in Armenian highland. So what they published within these 3 years, or what are their main results, is that majority of Armenian Y chromosomes, but it's not exact Armenian Y chromosomes, but at least it's collective term for all markers on Y chromosome, belongs to Neolithic haplogroups, so belongs to Neolithic age. It shows that our Armenian Y chromosome is
Very
here, starting from
Long time.
that time. There is also strong evidence that of genetic continuity of Armenians starting from the Bronze Age to our contemporary Armenians who live now in Eastern Armenia. They also identified during excavations a fact that Armenian highland was highly habitated starting from Paleolithic age. And they were able to recover bones and isolate ancient DNA, so DNA from bones coming from Paleolithic ages. And now they try to recover some genetic markers and then compare it to more recent genetic markers. We already have information about them. Again, to show whether we really were natural, like, habitants of Armenian highland. Yeah.
How old are these caves?
I think this one in Artsakh has about 30,000.
30,000.
Group of ecogenetics of populations tries to understand genetic diversity of Armenian fauna. They are specifically interested in black flies because black flies are very good indicators of environmental pollution. They studied different species that they were able to collect in Armenia, and using molecular genetic techniques, so-called DNA barcoding, they were able to identify 28 new nucleotide sequences in gene COX-I, and these new sequences were already submitted to genetic gene bank repository as sequences which are specifically come from endemic fly species in Armenia. This is more research related to biodiversity. Molecular neurology group is aimed to understand how structure and function of potassium channels in our organism can be related with diseases. If mutations that change structure or function may provoke disease development. They use a lot of different techniques. The technique I like most of what they do is site-directed mutagenesis, when they using different clones and plasmids specifically change nucleotides on the position they want to see how this affects the structure of the protein. And then they use electrophysiological techniques to record what happened after activation of these channels. So they find out that the mutations which are commonly seen in different diseases, like long QT syndrome. Have you heard about this syndrome?
What syndrome?
Long QT syndrome.
QT?
QT.
I have.
This is heart disease, QT. Yeah, it characterized by this. You remember the heartbeat pulse?
The
Yeah,
way,
it
yeah.
peaks. One of the peaks is QT
Oh,
peak,
yeah.
and it's very dangerous because even mild stress can simply cause heart stopping, heart not to beat. It's completely shuts down. And there are several mutations in these potassium channels which are linked to this disease, which are responsible for this disease development. And they were able to demonstrate that these mutations, specifically for the disease, are able to change the parameters of response for these potassium channels. And this might be a beginning for mechanism understanding. Karen Nazaryan, head of laboratory of computational modeling of biological processes, and his colleague are interested in so-called structural bioinformatics. Based on the physical evidence and knowledge about interactions of molecules and using more or less powerful computers, you are able to reconstruct how different molecules interact with each other. And why it's very important and interesting, because you can do screening for a lot of different interactions and then choose several potential ones and then test them experimentally. Otherwise, you need to do a lot of experiments. These methods allow to narrow the search field for, if you're interested in study drug protein or drug biomolecular interaction, these methods will allow you to narrow the field, choose maybe top 10 drugs, top 10 proteins, and then try to experimentally figure out which one is correct one. So they also interested in familiar Mediterranean fever. They studied this protein, pyrene protein, and they generated native full-size protein 3D model. And using this model, they're going to introduce mutations there and see how these mutations affect the structure of the protein and its interactions with other proteins. And this will greatly help to understand the mechanisms of this disease. Finally, my group. This is really my group. It was established in 2011. Yeah, I really, after having 10 years of wet lab experience, when you go and do all your experiments by hand, I decided to switch to data analysis. I realized that I like computers more than pipette, so I switched to bioinformatics, and mostly we work in field of genomic data analysis. So what we do, we develop different algorithms and software and use them for analysis of how genes are expressed in health and disease. We use them for analysis of next generation sequencing data and try to link this information with molecular networks to understand of pathomechanisms for various diseases.
Where do you obtain the data?
Data mostly we obtain from public repositories. There are a lot of data available. Now, data is not problem. Problem is correct analysis of data. Because you can have almost, the volumes become so big so you can even express the numbers. So at the moment we do have several data sets with terabytes of data. And the problem is to find out which instrument is correct for this specific data type and use it. Or develop, yeah?
Is that busy chart there on the right?
Which one? This one or
Which
this
I
one?
cannot read.
This or this?
The right,
No,
to my right.
I need to turn. Okay, so this is, I will slowly come to that.
Oh,
So what we do, we try to find if there are specific and shared mechanisms for disease development. So you know that there is something called autoimmune diseases. They can be very different in their clinical manifestations. They can be very similar, making them hard to diagnose. But we think that they should have some parts in their development mechanism, which should be very similar, but also they should have some mechanisms which are very specific to that disease. And depending on what you want to cure, for example, you can target the specific part or target the shared part. And we now work to understand how different pathways and their activities are changed in different diseases. We call them human complex diseases because they are not characterized by single gene change, they are not characterized by single mechanism, they are characterized by global interplay of different mechanisms affecting at the final point some important event. So, and another study which relates to this green strain chart on your right is a study of how telomere length is associated with age. Telomeres are caps on the chromosome ends, and they protect chromosomes from degradation. And when we become
Older.
Young.
I wouldn't say older, wiser,
Wiser.
Or under stress.
or under stress, making us more wiser, these telomeres become short. And there is a critical level of being them short. After that, cell stops dividing or even sometimes kills itself. It has also been shown that in cancers, these telomeres can be maintained on the constant length. And this makes cancer cells immortal.
Just a question.
Yeah.
But isn't this like genetically defined? What's the length of the telomere?
No. Actually, yes, no, I don't know. Why? Because there are no precise mechanisms. At the moment, we don't know any gene which is strongly linked to telomere length. From the other side, it is known that on the birth, girls have longer telomeres than boys. So it has some genetic background, but it's not certain like in case of familial Mediterranean fever. With this disease, you know, if you have mutations, you have disease. If you don't have mutations, you are healthy. With telomeres, it's not as straightforward. It's more complex. And another important thing is, depending on the length of telomeres, there are genes which lie very close to these ends of chromosomes. And when telomeres becomes very short, these genes start to be activated, and can work as sensors for cellular senescence. They can detect whether the cell becomes old or young. And we really are very interested in this. We call it telomere biology. And we developed special software to analyze this data. And now we use this software for an analysis of different data sets, different genomes, to find out the genes which are related to telomere length, and also find genes which are regulated by telomere length in health, in age-related diseases, and in cancers. These are just list of software was written by me and our colleagues in my group. All of them are located in public repositories, and I'm proud to say that we are not the one who use the software. We daily get a lot of downloads from worldwide, actually, mostly from China. I don't know why, but we have also significant downloads from US and Europe. This part is just few slides showing our activities related to education and professional development. As I told you, this is a very important part of our activities, and we constantly try to involve as many professionals as possible. So if someone comes to Armenia, even for a short-term visit, we try to catch him, bring to institute, and make him to deliver talk to us. And we also organize practical courses for young scientists, and we usually invite students not only from our institute, but from university, from the Slavonic University, from other research institutions. So this is slides from our practical workshop on functional characterization of potassium channels. We have invited guests from Germany, Professor Jürgen Schwarz, and our head of group, Vitya Vartanyan. It was 4-day course, and it was both theoretical and also practical. So they physically sit in front of the lab bench and did their experiments there. And as you see, they were very happy. And actually the guy here, again, you can't see him very well, he was from Spain. Somehow I don't know how he was able to find our announcement, but he just wrote us that I want to be there. I said, welcome, and he really came, which was more. We traditionally organize schools, young scientists schools, which is called post-microbe interaction. It's maybe 6 years. We have, yeah.
How old are these students?
So when we call young scientists, these are starting from 20 years to 35. And our very good collaborators, Rafi Aryan, Bana Jabri, and Haik Iskandaran. Rafi works at the University of Massachusetts. Bana Jabri works at the University of Chicago. Haik is now postdoc in Lausanne Polytechnic Institute or school, École Polytechnique, and they come each year and may deliver lectures for young scientists. They work with them on, they discuss paper with them, they do practical courses, and in 2015 we had the 4th, in 2016 we had the 5th. Again, it was very good 3 days, it was very intensive work, and I should say that these activities are really very important for us, and our students really learn a lot how to write papers, how to read papers, and how to deliver speech to audience, which is essentially another important things to learn. This is practical course organized by my colleague, Henry Wirtz. He is from Leipzig University. He also is bioinformatician. And we asked him to make 4-day course for bioinformatics data analysis and programming. So we put 20 people in one class, closed the door, and they were there for 8 hours per day. So they were completely exhausted at the end, but also happy. And these are, as I told you, we always invite someone to deliver, like to have some small talk or workshop in the institute. These are our new colleagues. We just get acquainted with them maybe 6 months ago, and they were visited us and delivered lectures at the institute. This is Professor Ardem Pataputyan from Scripps Research Institute. He also a neurobiologist, and we were happy to host him, and then they had a lot of interesting discussions with Vitya Vartanyan. Virginia Zakyan, a famous researcher in field of telomeres. Actually, she and Elizabeth competes in this area of research. We were really happy to host her at the institute, and she delivered a beautiful talk about telomeres, and then even more beautiful talk about her origin and her family and her heritage. Our youngsters or young scientists are, it's actually what we did for them should some less or soon shows up, and it really starts to show up. Our young students have won a lot of awards, both on national level, on international level, and these are just few which I was able to find from my computer. So Renato Vanessian, now he's a postdoc at in Barcelona. He got the President's Education Award. Lilith Nersisyan is awardee of ARPA. For ARPA, she has got ARPA Innovation Award. So I'm 37, so I'm not considered as a young scientist, but me and my colleagues won award Most Productive Research of 2012, awarded by State Committee of Science of the Armenia. We constantly send our scientists to different conferences, and they constantly bring back certificates for best oral presentations from everywhere. Even now, I dedicated one poster for that. Now we have 2 of them there, and I really hope that soon the year will end and I can free these posters from these certificates to open space for new ones. Of course, everything couldn't have happened without our scientists, our technical and administrative staff, our partner medical organizations that are very interested in collaboration with our institute, and they are really willing to contribute to the research. Patients and healthy volunteers that agreed to provide us with their samples. Our funding agencies and donors, and ARPA, and also our national and international collaborators for their continuous and strong support of our activities. And thank you very much for being here.